Yes, yes.
Okay, the title of the presentation is Determining the Optimal Biomarkers for Assessing Mineral Status in Elephants using Zoo Elephants, and the presenter is Fiona Sach.
Hello, good afternoon everybody. I'm Fiona and I'm from the University of Nottingham and the British Geological Survey in the United Kingdom, and I'm a PhD student and I'm looking at determining optimal biomarkers for assessing mineral status in elephants, and I've been doing this by using a population of UK zoo elephants to help to validate these methods. And I'm looking at this because I'm looking specifically around the Palabora copper mining complex in South Africa, where there has been some quite considerable human-elephant conflict. The elephants are coming onto the mine, as you can see in these pictures here, and causing disturbance. It risks human life, elephant life, and also causes financial loss to the mine, as when these situations occur, the mine has to close, which causes a financial loss of about R1 million a day. It's particularly unusual behaviour for an elephant because this mining area, as you can see, is very busy, it's very industrial, and it's not a sort of typical environment where you would hope to find an elephant. The workers of the mine understandably don't want the elephants to be there. They have a lot of fear around them, a lot of discomfort, and generally it's not a very sort of amicable situation. So this is particularly important because although this applies to one situation here in South Africa, just to the west of the Kruger National Park, it's hoped that these methods that I'm using to look at this could be applied to other situations, other similar situations of human-elephant conflict, really internationally, and really to see if minerals are affecting the movement of these elephants and if the provision of minerals could be driving this movement pattern. So this is just very briefly my study site. I'm sure everyone here is familiar. But the Palabora copper mine is just to the west of the Kruger National Park. It's an absolutely enormous open pit copper and phosphate mine, and if not the oldest, certainly one of the very almost oldest copper and phosphate mines in southern Africa. Mining activity has been going on here since about the 1950s, and this sort of elephant movement has really been observed in the sort of last 8 to 10 years, causing particular conflict there. So as you can see here in this picture here, this is an aerial photograph of the mine, and this is collared data from 7 collared animals collected over a 5-year period. And the pink dots on this map here show the movement of the elephants. And what's particularly concerning is these elephants are, as I say, coming directly onto the mine site up here and drinking the water and spending time in this water here, which is sort of runoff water from the mine. So this is a real concern as well for the elephants, because they could potentially be being exposed to quite high levels of trace metals, which potentially could bioaccumulate within them and cause an issue. And this just shows the scale of the mine and the sort of situation that we're faced with. So there has been one security guard that's been killed at the mine by an elephant, and there have been 3 elephants that have been killed as damage-causing animals, which we heard about earlier today at the mine. So generally it's just not a very positive situation. So this here shows the home range of these elephants. This purple is the entire home range of this population of elephants. And the red dots identify from a previous study areas that were identified, soil samples that were identified as having quite a low phosphorus content. Now this is to be completely expected in African soils, but it just gives some evidence to the hypothesis really that it could be a mineral that's driving these animals to spend more time up here in these sort of higher phosphorus areas compared to the surrounding areas. So we'll come on to show some data a little bit later, but that's just the kind of background to the situation. And the home range for these mine elephants, as you can see here, is considerably smaller than documented home ranges in the literature of other typical Kruger National Park or associated private nature reserve elephants. So the real aim of my study is to investigate how environmental geochemistry affects elephant movement, and I'm doing that through the objectives of establishing baseline levels for key minerals and trace metals in African elephants, so really establishing what normal is, determining if mineral levels in soil, forage and water around the mine are considerably different to the surrounding area, and determining if the elephants near the mine have higher essential minerals and or trace metals reflected in various biomarkers within them. So I validated this study. I did a year-long study with 5 UK zoos that I visited 4 times, and the nice thing about sampling in a UK zoo is you can control most of what goes into the elephant and have a reasonable idea as to what's coming out of the elephant. So I had various inputs that I decided to sample, environmental and diet, and then I had various outputs from the elephants, so plasma samples, fecal samples, tail hair, toenail, and urine samples. I did all my sample analysis on an ICP-MS 8900 at the British Geological Survey, and I've really tried to focus my work around producing good data. So for every 40 samples I've run 3 blank samples and 3 reference material samples to really make sure that the data that's being produced from this work is really, really true. So these are the 5 UK zoos that I've been working with, and they were selected based on their geographical location to hopefully show some differences within the soils which the elephants are exposed to. And this just shows the samples that I collected, so the input samples and the output samples over here. And then these were the samples which were more difficult to quantify, so the amount of grass that these elephants are eating, the water that they're drinking, and the soil that they're exposed to. I had African and Asian elephants in my study, male and female, 22 elephants at the beginning across these 5 UK zoos. So this just shows the diet and the water compared to recommendations published in literature. And what's particularly interesting here is generally our UK zoos aren't mineral deficient compared to the recommendations. But zinc came up as a specifically interesting one because we had 2 zoos that were slightly below the recommendation, and one of which, when we consider the water that they were provided with, their levels really rose. And that's something we rarely do. And when these elephants are drinking up to 200 litres of water a day, it's very important that we do actually consider the mineral and potential trace metal contribution that this water can make. This is just some of the outputs from my zoo elephants that just shows that they're in line with the levels in the literature from both captive and wild elephants. These recommendations in the orange boxes were taken predominantly from Species 360, which is the database which maintains this information across all captive samples that are submitted. Now we have to remember some of these animals could be immunocompromised, and the sample sizes can be very small. So it's not 100% ideal, but it just shows that things were vaguely in line with what we'd hoped, which is good and gave me some confidence in my analytical techniques. So I decided to do a linear mixed model using R and the LME function, and I combined the input that I had of the grass. I made an estimation as to how much grass they were eating based upon total dietary input. I estimated how much water they were drinking based on the literature and recommendation of 200 litres of water. I also had their very accurate keeper-fed diet, so I had a very accurate idea of what they were being presented each day in terms of their hay, browse, pellets, fruits and vegetables within the zoo. So I had that information that I was very confident in. I was less confident in the grass. So I took all of these and put them into the mixed model, and as you can see there was some significance which came up, which was great. I then decided to look at what the most likely contributors for the significance were. So I did it sequentially with diet, then grass, then water, then soil. And I applied alpha wealth to make sure that I wasn't falsely accepting the hypothesis, the null hypothesis, and to make sure I wasn't through the chance accepting something that wasn't really true. So alpha wealth knocked out 11 significant relationships, which is really good because it increases the validity of the data that we're producing. I had fixed and nested effects in the model. I looked at sex and species, zoo, the individual elephant, and the season, because I visited each zoo 4 times throughout the study period to account for seasonal variation. So in conclusion, we really showed that no single matrix is suitable to monitor mineral status in elephants. So historically and classically, we take blood as the best sample, and it just really shows that maybe we need to be looking a little bit more broadly at this. The macrominerals are not reflected very well in plasma, and this is completely to be expected because the body will be homeostatically controlling that, and that's completely what we would expect, but it just really confirmed that. UK zoo elephants are generally not mineral deficient, which is quite positive. But UK zoo elephants are vastly eating over the 1 to 1.5% dry matter of their body weight per day, vastly over this. So this can probably indicate some of the obesity levels that we do see within some of these captive animals. So applying this to the wild, I was working with partners Elephants Alive and the South African National Parks, and I had collared elephants that were at the mine and outside of the mine, and bank samples from the South African Biobank. So these are the samples that I had. Unfortunately, we didn't have any urine samples, but that was not a problem. And these were my sample sites. So I did sampling from 2016, 2017, and 2018, and this is the mine here and here. So I did intense sampling around the mine, and then about a 50-kilometre radius outside of the mine, going diagonally, as you can see on these these yellow spots here, and the red here just shows where the previous study was done, and the purple and the blue show the home range of these elephants. So we sampled soil, water opportunistically, and plant samples at each of these sites. And the plant samples that I sampled were the 6 most commonly consumed species within the area, found within the literature, and talking anecdotally to people that seemed sort of sensible to be sampling. Also, I sampled fecal samples opportunistically at each site, and must obviously be remembered that a fecal sample at the mine doesn't necessarily mean the elephant was consuming at the mine at that time, but it was just another indicator that we decided to include. So this is looking at the phosphorus in the soil against the mine and the non-mine, and as you can see, we do have some really high levels of phosphorus within the mine soil. It's completely to be expected that we would have some levels that are overlapping because these two places are very, very close to each other. But we really are seeing these extremes in high phosphorus, which is really positive. And this is the water, and I've just chosen these elements because they came up as being the most interesting. But again, we are seeing these very big differences between the mine and the non-mine water, which is great. These are the fecal samples, and again, we're seeing some really good differences. We must remember that, as I said, fecal sample at the mine doesn't necessarily mean that that elephant was consuming at the mine, but it's a good start. The model didn't predict phosphorus, fecal phosphorus being a very good indicator of phosphorus level in the elephant, but I've included it here because we are seeing a difference, and we saw such a stark difference within the soil. And then I was just looking at our zoo samples compared to free-living elephants to see if we are seeing any differences. This project is very collaborative, and the idea is it's a good opportunity for zoo elephants to benefit wild counterparts, and potentially wild counterparts benefiting from the samples and the validation process that was able to happen due to the zoo elephants. And what we're seeing is that the zoo elephants don't particularly look to be, the zoo elephants are here in the green, don't particularly look to be lower in selenium, which is really positive. I've just sort of picked this as it was an interesting one to include, and selenium is something that we do worry about in our UK soils sometimes, particularly in certain areas. And so that was a really kind of positive start. I'm only comparing to African elephants because although I didn't find significant differences in the model between African and Asian, I don't think it's sufficient to assume that all of African and Asian will be the same. We must remember that the zoo samples were selected very specifically from a very predetermined sampling strategy, and so statistically we probably can't compare these very easily other than visually. So it could suggest here that some zoos are feeding quite high levels of selenium, and that's why we're seeing it reflected in these matrices. Just to finish, this is something I've been working on that's been quite exciting. So as I was doing my tail hair analysis, I generally did sectioned tail hair analysis, so took 5 centimetre or 3 centimetre sections of the tail hair and analysed that through ICP-MS to look at the mineral levels. And it kind of crossed my mind that we don't really know how mineral levels change cross-sectionally across the tail hair, and whether there could be any variation that way, and also whether we want any more specific, really detailed information about how that mineral changes down the tail hair. So I used laser ablation, which takes a really tiny sample from the tail hair and still leaves the tail hair intact so that you can use it for full mineral analysis. But it just gives you a very small number of elements that you can sort of whiz a laser down, and it's quite quick, so it can give you quite an initial view of how that mineral status of those specific elements could be changing down the tail hair. And as I was doing that, it kind of struck me that we don't really know how deep this laser is going into the hair and whether cross-sectionally that could change. So this photograph here is a scanning electron microscope photograph of a tail hair with a laser trench, and you can see that it doesn't really go all that deep through the hair. So that was what made me really think we need to see what's happening a little bit cross-sectionally. So I lasered cross-sectionally, so I took a slice of the hair, mounted it in resin, and then this black line here is where I lasered cross-sectionally. And this graph just shows that we do have clusters of higher mineral levels at the edges of the hair, and this is what's seen in other mammalian species. So it's just quite nice to confirm that that's happening in elephants. And this is just a normal microscope photograph that just shows how the laser whizzes down the hair, because it's quite exciting. And this can give you a lot more information about where you do your sectioning to make sure that you're really maximising these really, really precious samples. So really, to sum up my talk, the aim is to potentially reduce human-elephant conflict around the Palabora mine and identify if these elephants are making this movement choice based on mineral need, and I guess the part two would be whether you could provide these minerals, whatever this mineral is, in an alternative form to pull them away from the mine. And it's to benefit free living and captive elephant research and to have a really good collaboration between the two. It's been a very multidisciplinary project, and so I'd like to thank all my supervisors, Centre for Environmental Geochemistry, my PhD is a NERC-funded Envision DTP, Elephants Alive for all of their samples, the South African National Parks again for their samples, and from the very precious samples from the South African National Parks Biobank. I've also been working with SEON, and Save the Collars have been done by Save the Elephants. So thank you very much. If anyone has any questions, I don't know if we have a quick second, or please feel free to ask me at a later time.